It is my pleasure to introduce Ryan Phelan. And my day job is I'm a long-range forecaster, and so I pay attention to people who are good at forecasting. And Ryan has this absolutely unerring ability to see emerging connections way ahead of the crowd, way ahead of the pack. And what I've watched is the first time she'll mention something new, everyone go, that's kind of surprising and interesting. Then after a while, everybody says, about a year later, now I start to get it. But then eventually that turns back to surprise, because when you realize what you think you got really was only part of the story, and Ryan was doing something much deeper and much more surprising. So all I can say is I invite Ryan up to the stage, prepared to be surprised. Ryan?
Thank you. This is a painting by Isabella Kirkland, an incredible artist who's actually in the audience. Its title is Back, and it depicts 48 species that were all brought back from the brink of extinction. This happened just at the end of the late 1990s, and it was done using traditional tools of conservation. That means these species were brought into captivity if needed and be rewilded over time. Their habitat was protected. They stopped overhunting. These are all the common tools of conservation. But now, as we enter into the 21st century, we have new conservation challenges. We have invasive species like never before. We have novel wildlife diseases. We have smaller and smaller populations of endangered species losing genetic diversity. And all of that is compounded by climate change. And everyone in this room knows that the world is warming. And even the latest projections show that it's going to get way worse, and we're going to lose biodiversity along the way. Now, my background actually is in healthcare. I spent several decades in it, and I saw the Human Genome Project when it was launched. And I knew it was going to be transformative for healthcare. I could see the emerging view of personalized medicine, where we could have targeted therapeutics and precise diagnostics. And then we could see this happening with precision agriculture as well. And the foundation of that biotechnology platform really begins with genomic sequencing, high-level sequencing and bioinformatics. And continued with advances in cell culturing, advances in reproductive techniques. And we could see at the time that genome editing was coming on the horizon quite fast with precision gene editing. But this platform had not yet been applied to conservation. And my husband, Stuart Brand, who's a co-founder of Revive and Restore, we were both thinking 10 some years ago, what if these tools could be used to help solve some of the emerging problems in conservation? And for those of you who know Stuart and the Long Now Foundation, you won't be surprised that he opined, well, you could probably even use these technologies to bring back an extinct species. And the passenger pigeon became one of our flagship projects. We started within the Long Now Foundation, where this idea was incubated, and Stuart and I launched a series of meetings with scientists from around the world. We brought in genome engineers, social scientists, and ethicists to weigh the benefits and the risk of these kinds of projects. And it became very clear that what we needed to do was actually start to socialize these ideas, that these tools were going to actually be used in conservation. And with that, we coined the term de-extinction, and we took it to a TED stage. We brought 18 scientists that were actually doing genome editing and beginnings of de-extinction work all over the world. We brought them to the stage where they could hash out some of these issues at National Geographic. All along the way where we were creating this, we could see that these tools could be used to help endangered species. And with that, we actually named our organization Revive and Restore to keep our eye on the prize, which was that it was really not just about reviving individual species, but it was really about ecological restoration. And the first chance we had to work on a project that was really so profound for our organization was with the black-footed ferret. We got a call from Fish and Wildlife, and we heard the story of the black-footed ferret. It used to range across the Great Plains from Mexico to Canada. And that was until in the 1800s, this land actually became agricultural fields. And the black-footed ferrets and their primary source of prey, which is the prairie dog, were all literally exterminated. And by 1981, there were none, until one small population was found in Meeteetse, Wyoming. Wyoming Game and Fish brought that small population into captivity, bred them over time, and very successfully have reintroduced them to over 30 sites back into their original range. However, over 30 years, all 10,500 ferrets have been bred in captivity from just a founder gene pool of 7 ferrets that had been wild caught. Now, everybody knows that inbreeding is not a great thing. And when species get into smaller and smaller populations, they run the risk of going down the extinction vortex. When smaller populations cause greater genetic drift, lower fertility counts, and even become more susceptible to wildlife diseases, increasing their mortality. Now, when we got the call from Fish and Wildlife to ask if we could help, the first thing we said was, is there any other population that you can bring in to restore genetic diversity? Because that's the typical tool in technology. Bring in a population from another area and increase fertility, lower the mortality rate, increase the population, and becomes more stable. But there was no other population for the black-footed ferret. So we heard that within the San Diego Frozen Zoo, back 40 years ago, somebody was prescient enough to actually bank cell lines from 2 other individuals that had been wild caught in 1981, but had never contributed to the gene pool. With that, we did the genomic sequencing on those 2 cell lines, and we found that they carried up to 3 times more genetic diversity than any living ferret. So if we could actually clone those ferrets, we could start to build up that population, not by moving animals, but by moving genes. By partnering with the San Diego Zoo and a partner, Biogen, as a cloning company, and with U.S. Fish and Wildlife under their permit, in 2020, we cloned Elizabeth Ann. She's the first endangered species in America to ever be cloned. Now, this is an amazing success story. And it was a first for conservation to actually use these technologies in this way. But to me, what's actually almost more amazing is the fact that scientists back in 1980s were prescient enough to start to think about cryopreservation. Now, cloning didn't exist then at all. They didn't know what they were going to do with these cells. And that got us thinking, what else has been ever cryopreserved? It turns out that less than 14% of US endangered species have any form of cryopreservation. That includes plants and animals out of the 1,500. That's an alarming low number. And it's a particularly alarming low number when you realize the rate of extinction loss just in the last 100 years, and the trend keeps going up. So with that, we started talking with officials at Fish and Wildlife Service, and we suggested to them that if we could figure out why this isn't happening, that we could actually help create protocols and actually a roadmap to help ensure that every species that is endangered in the US, and the Fish and Wildlife Service has a responsibility to actually get them back off the list. We've said, why don't they just include in that whole process taking a cell culture and ensuring that that species has a reference genome that can be open access and used by researchers all over the world. So with this idea, we're trying to actually bank species for the present to help make better management decisions based on their DNA. But the truth is, they're really going to be put in the bank to also help future generations with unknown technologies that are unthinkable today, but could be transformative in the future. And here's our partner, Seth Willey. This is actually the same guy who called us to ask, could we help him with the black-footed ferret? 10 years later, Seth is a major partner in this whole initiative.
We're responsible in the Southwest region for about 200 threatened and endangered species. Nationwide, we're responsible for about 1,500 threatened and endangered species. And it got me wondering, are we doing our part to set up future generations the same way somebody set up black-footed ferret for that success 30 years before, by biobanking, by cryogenically preserving that specimen, that sample? And the answer we came to was hit or miss. We're doing it in little bits here and there. In conservation, if one of the first premises is save what you have, this is one tool that gives us that option to save what we have, not for academic purposes, but for recovery, for genetic rescue, and push species back to the point of being biologically viable. In this game, in conservation and endangered species recovery, we're entrusted to protect some of the nation's most valuable and unique biological resources. That conservation legacy is entrusted to us, and we're responsible for trying to arrest a species decline, to manage threats, and to bring species back from the brink. Genetic diversity took millions of years to evolve, and we're losing it on our watch. But it's not too late, right? There's still great opportunity for those critters and for many others. It's our responsibility to try to do what we can to save what's there today.
So that's what we're doing. We've started this pilot with our first species. That's the Mexican wolf. It is native to New Mexico and Arizona. There are only 200 left in the wild today. And on a routine vet check last month, the field biologist team, working with Revive and Restore, actually was able to sample that Mexican wolf, send off the sample to our culturing partner with Viagen, and it's now on its way for sequencing and will be going for long-term storage at a federal facility. So one down, we've got another 1,500 to do. We've talked about cryopreservation with mammals, and we started there because really, to be quite honest, they're the easiest to do. But for many organisms, it's incredibly hard. And we're working on some innovative research to actually try to figure out a solution to that. But probably nothing is more important right now than working with coral. There are over 800 species of reef-building corals, and coral reefs play such an incredible role in ecosystem. They support fish, mammals. They also provide incredible support to humans. They protect our shores, and they create the livelihood for over 1 billion people. And we're losing coral reefs at an alarming rate. Projections say by 2050, we may lose 90% of the coral reef from bleaching. Now, bleaching is not always 100% extinction, because if coral have a chance to rebound the next year with cooler waters and less acidification and no fishing trawling on them and everything else, if they don't have repeated insults, they can actually come back. And so we need to capture that biodiversity before it's really gone so that we can actually help bring those reefs back to fruition. And we're working right now with a project with Mary Hagedorn, a leading scientist at the Smithsonian. Let's hear from Mary.
I've been working on the cryopreservation of marine species since 1993, and I realized it was going to be important because you could tell the oceans were going to be in trouble from climate change. Initially, when I started, I was really just trying to work out the science of how we do this, but then it became very clear that things were changing very rapidly, and climate change and bleaching events were happening, and I became very worried that we needed to get this science done quickly. I work on the conservation of coral, and I use human fertility techniques to do this. Basically, we are cryopreserving or freezing, however you want to think of it, gametes, the sperm and the larvae of coral, as well as small fragments of coral. And the reason we are interested in using cryopreservation is that it allows us to stop time, really. We can freeze coral and keep them for a day in our bank, or 10 years, or 100 years, or 1,000 years. So it gives us the ability to use this material today or save it for many, many years in the future to potentially reseed the ocean. One of the major outcomes of our process is scalability. We want to be able to cryopreserve coral easily and cheaply so that every professional that wants to around the world can form their own cryopreserved banks of coral and thus speed up the process of securing the biodiversity and genetic diversity of coral.
So Mary's project is one of 8 that Revive and Restore is funding right now. We call it the Advanced Coral Toolkit. And we have researchers working at understanding the underlying genetics that allow some reefs to be resilient and continue in the warming waters, some that may even show signs of resilience to disease. So as these scientists are working, they're developing the ability to create coral stem cells that could be used to repopulate reefs, as well as new techniques using proteomics and other parts of research to figure out how to combine these tools so that we can actually make a big difference when we can make an effort at restoration. Now, we've talked about cryopreservation for a bit here, but I want to make it really clear that one of the most simple and what should be a ubiquitous tool for conservation is genomic sequencing, that foundational level for everything. And as sequencing costs have plummeted over the years, this is a tool that I just wish more conservationists could actually access. And we're doing our effort to try to make it more ubiquitous. We have 2 projects I want to talk about that I think are really exciting, just in the way that they're using that genetic insight to make better management decisions around these endangered species. And the first one takes us to Kenya with an Irishman named Andy Tai, who's been doing some incredible work.
Arabuko is the largest remaining remnant of coastal forest in all of East Africa. And it has a high degree of endemic species. And there's a population of elephants that are currently living in this forest, which in the past would have been connected to other populations of elephants, including those in Savo and further north and further south. But now they're surrounded by an electric fence. Because it's such a dense environment, it's been very difficult to actually count how many elephants are in there. And it's very difficult to manage a group of any animals if you don't know how many exist. Where this project is kind of pushing new boundaries is that this will be the first time a SNP array has been developed for savanna elephants. SNPs are single nucleotide polymorphisms. So basically one base pair in the genome where individuals differ. And when you amalgamate all those differences across an individual's genome, that pattern of SNPs is unique to the individual. So by using this approach, we can see how many individuals are in the forest. And the way that we're doing that is non-invasively using scat. In Arabuko, we have a unique situation where in the dry season, all of the elephants in the forest are forced to visit one watering hole. And what this means is that we can get a snapshot of every elephant in the forest. As it comes to drink, it will relieve itself and leave us a sample. And as we sample over the course of 2 or 3 weeks, we should in theory cover the entire population. Knowing this, wildlife authorities in Kenya will have a clearer idea of the pressures that are on the forest. And if the number is quite high, it will indicate to wildlife authorities that perhaps some elephants should be translocated or a wildlife corridor should be put in a way almost like a release valve. But a unique part of our project is that we are helping to develop a local lab in Kenya. And by having it based in country, it means that these sorts of projects can be happening with much higher frequency and are reliant on people like me flying in, facilitating stuff like this. Ideally, in the future, human civilization will have space for nature, but at the moment it's a fight. So areas like Arabuko, Sokoke need to be protected in their entirety, which includes the elephants and all the other animals that are in there, so that in the future, when we have space and tolerance, they'll be able to spread out. This is true of all wild places around the world.
What I love about that project is that these elephants are absolutely invisible from the sky. So they really don't know the size of the population, and they don't know the level of inbreeding that could be happening in a closed population. So by looking at that individual fingerprint of those animals, they not only know their genetic diversity, but they will begin to even understand what plants they're eating and how they might even create a migratory path for them. Now, using genomic insight has a very different take in a very interesting situation close to our home here in California. This is a healthy kelp forest, and this is the way it used to be from Baja California to Alaska. Rich, diverse ecosystem, creating the habitat for millions of organisms and nurturing mammals along the way. Now, what creates this healthy ecosystem is the sea star. There are 8 different species of sea stars, all up along the coast, or they should be. And they got hit very hard by an unknown invasive pathogen, and it's called sea star wasting disease. And it absolutely disintegrates these sea stars. Now, the billions of sea stars used to be here until 2013. And what is replacing them is all purple sea urchins. It's been identified that we've lost 95% of our kelp all along this west coast. It's a horrific number. These are the bull kelp, the ones that should be like a forest. And these urchin barrens are terrifying, particularly because they can last for up to 50 years. They just go dormant. It's a crazy, horrible situation. And right now, there's a lot of different ideas of how to remove these urchins. There's an urchin barren that can be seen that is 400 miles long off our coast, from Marin County to the Oregon border. There's lots of technology, but there's one technology when it was proposed to us for research that we thought biotech might have a solution. And I'm going to let Mike Dawson tell that story.
The question for us was, is there a way to reintroduce the sunflower sea star to California, Oregon, and Washington, and to help those populations that are reintroduced be more resilient to future change, right? Whether that's changes in ocean temperature, ocean acidification, or other outbreaks of wasting disease. And so the way we approached this essentially was to try to work out what diversity was remaining or what diversity had survived after the wasting disease. And having identified those portions of the genome, we're developing a way to rapidly assess the diversity of genomes to identify which animals should have a tendency to be resilient to wasting disease. We're now collaborating with a team in Washington to use the information that we have about the genomics in a breeding program to restore populations in the wild. With the Nature Conservancy, we're now actually collaborating to try to do that part of it. The next part of that project will be to take the tools that we're developing with Revive and Restore into that actually more, real applied setting that has been the long-term goal. We've become more willing to try technical solutions to conservation problems, in part because we've just learned a huge amount more about marine systems in the last several decades. Over that same period of time, 2 to 3 decades, we and the general population become much, much more aware about how severe the current threats actually are. And so the need for action. And so the idea is that by restoring populations of sea stars, we would also be able to help build the resilience of kelp forest ecosystems.
So I think this is a great example of biotechnology being an interesting part of the solution. But let's be clear, it's not going to be the magic fix. It's going to take all kinds of innovation and technology, from robots to aerial surveys to, divers actually removing them in great quantities. But it's the combination, it's the integration of these technologies that is starting to happen that is exactly why we started this organization. But let's talk about birds now. 1 in 8 species of birds are threatened globally with extinction. It's an alarming number. Half of them are in decline. Hawaii has lost 75% of their endemic species. Some of that is due to climate change, habitat loss, but it's also due to avian disease. We've been trying to figure out how to use some of the technologies that we're using in mammals, as in cloning, to actually influence and help solve some of these challenges with birds. But the problem with the bird is it has a very unique reproductive system. The embryo is tucked inside the egg, and the egg is a huge barrier. Mammals with uteruses are actually easy to manipulate, it turns out. And what it's going to take to change and actually develop advanced reproductive technologies for birds is different. It's called germline transfer. And it's actually been done successfully in the poultry industry with chickens. And what we're trying to do is learn from that technology and to apply it for endangered birds. We've just announced a program called Biotechnology for bird conservation, and we have 8 international teams working on this exact problem. They're taking different paths. Some of them are incredibly synergistic, and some of them will be sequential, but all of them are working together to actually solve this challenge. The way it would be done is you would take those primordial germ cells from the endangered bird and you would transfect them into a domestic surrogate, and the offspring would actually be the wild bird, just like we did with the ferret. If we could do that, you could actually repopulate some of these small, limited, endangered populations, because those common birds could actually have multiple chicks and multiple times a year. Whereas the endangered birds, if they're lucky, they get 2 or 3 chicks a year, maybe 1 or 2 survive, and you can't rebuild a population. I'm going to introduce Eric Jarvis, one of our scientists that we're working with at Rockefeller University.
My main line of research is understanding brain mechanisms of vocal learning, the ability to imitate sounds like we do for speech. And there are only a few species that have this ability besides us humans, like songbirds and parrots and whales. And as I started to get involved in comparing the brains and genomes of multiple species, I started to realize that a number of them I wanted to study are critically endangered. And that got me more involved also in conservation, because I feel like it's a moral duty for us scientists to get involved. Maintaining bird cells, the germ cells that is, for whatever reason, is harder to do than the equivalent cells for mammals. There's something about bird cells that they need to be inside the egg in order to develop properly than in a culture dish. My lab and many other labs are trying to figure out what is the magic sauce that these bird cells need that we've already figured out for mammals, but not for birds yet. And when we think once we figured out for birds, it's going to be the same for reptiles, because the birds are basically reptiles and they have the same egg-laying kind of system. A lot of folks think that conservation should be only about trying to put fences up to save species from population decline and not really get into the biology of, diseases of these animals or the genetics and so forth. I often have to explain to conservation-minded folks that putting up fences is not enough. With small population size comes the prevalence for more susceptibility to disease. Human population growth is going to continue to grow. We must find modern means to live in coexistence with species without causing their extermination. And so for my efforts in conservation, I'm doing them not only because I'm fascinated by the science, but also because I feel like it's necessary. It's necessary for our survival.
And the truth is, if Eric Jarvis and the other scientists that we're working with are actually successful, someday we might even have back the passenger pigeon. And when we do, there could well be another iconic species that was lost, and that's the American chestnut tree, and it's coming back. There were billions of these trees all across this range. By the 1850s, a blight came in from the landscaping trade that absolutely decimated these trees. And all that's left is just the ghost of those roots. The good part of the story is that for the last 30 years, there have been scientists at State University of New York that have been trying to figure out how they can create a chestnut tree that is blight resistant. And they've succeeded. It's really incredible what they've done. And they've done it with one transgene from wheat. This is Bill Powell. He's the scientist who has pushed this forward. Now, his team has had to go through incredible hurdles to bring this tree through the regulatory path because it is the first genetically engineered wild species. And he's really been a model for revive and restore. And the part that I particularly love about his story and why we didn't have to fund them, all we had to do was just advocate for the tree, is because he actually was transparent about the research that he was doing from the beginning. And I believe this year is the year that these trees will be commercially available. The chestnut is not technically our project. I just am a huge advocate for it. And as I said, it's been a model for us for all these years. Now, Revive and Restore has been involved over the last 10 years on 54 projects. And we've been funding projects in 16 nations. And those projects are focused on 70 wild species. But wild species don't live in a vacuum. They require healthy ecosystems. And those ecosystems include people, their culture, and their values. Now, we were really excited when we heard that there was a project called One People, One Reef, operating in the middle of the Western Pacific in Micronesia. And what excited us about this project was the fact that it was trying to combine traditional knowledge with modern science, with indigenous people. Let's hear from Magul Rulmal.
We're from the atoll of Ulithi. It's part of the Yap state in the chain of what we call the Outer Islands. And it's basically about a stretch of 99 islands in Micronesia. We're very subsistence community. We depend so much on maybe 90% on our resources, our local resources. So we still grow our own food with very little import. I think like many people, many coastline communities, we are facing climate change. And that's no surprise to everybody, but definitely not for us. We used to be able to predict certain birds and certain fish migrate associated with these weather patterns and those associated stars that we call the traditional markers. Now, these patterns have changed quite a bit, and we can't depend so much on traditional knowledge because it's changed. And so I think that acceptance of that and that willingness to seek further or new information kind of started earlier on. We need to seek more information because it's moving much faster than we ever know. So it takes science, it takes, genomics and DNA that is now collected to the way that more accurate count of assessing the reef health and the fish count. It takes that kind of science and knowledge to be able to help us make a decision on management. We've seen positive changes in combining these wisdoms of the West and traditional knowledge and coming up with creative ways to adapt to the fast decline in some areas. I think that the responsibility now in my mind is to spread this to the region on low-lying communities like us to join the efforts and see from the 10 years of data of how we can and we will be able to fight some of these fast-changing decline in our reefs and resources.
Now, the challenges that Magul and his people are facing may seem very remote to everybody sitting in this room. But I suspect that most of you don't realize that we actually do impact endangered species in ways that you may not even think of it. This is a blue bottle. It's actually called blue blood. And probably everyone in this room has benefited from this substance. It's harvested every year from horseshoe crabs. 400,000 crabs a year are harvested on the East Coast. They're impaled in these factories where 1/3 of their blood is drained. And the reason that you've benefited from this is because of that blue protein. It is used for every injectable drug or vaccine that you've ever had or anyone you know to make them safe from the impurities in the manufacturing process that could come in from bacteria in the water. That LAL substance it's referred to is the gold standard for drug manufacturing today. And it's used with not only drugs and vaccines, with every implanted device. And it's used even in veterinary medicine. But here's where the horseshoe crab lives. This species comes in once a year on the high tides of May and June to spawn. And it spawns in the thousands of eggs every year. And it provides not only for the proliferation of its species, but also as sustenance for wild birds and a whole ecosystem. And these wild birds, in particular, this is the red knot, they migrate all the way from the tip of South America all the way to the Arctic Circle. And they make one pit stop in Delaware Bay to refuel on the eggs of the horseshoe crab. Now, these species are being listed. They're endangered. They're not making it to the Arctic Circle, and when they're making it to the Arctic Circle, those that do are so thin and underfed that they usually are not breeding. This is a multi-continent ecological problem, and it's something that is done for the welfare of humanity. That's why it's so linked to everything that we do. But it doesn't have to be that way. And that's where synthetic biology comes in. Scientists back in 1995 at the University of Singapore, Dr. Ling and Dr. Ho, actually identified that unique protein in the horseshoe crab that they make the LAL substance from. And that unique protein was synthesized and has been created what we call the recombinant factor C. That RFC has languished on the market because of corporate greed, because there's so much money in the blood of the horseshoe crab. And it's also because of bureaucratic inertia and regulatory inertia. We've been advocating for the pharmaceutical companies to do something, to adopt this synthetic alternative, to force the regulatory authorities to do something. We and other conservation groups have organized a coalition, and we've sent letters ad nauseum. But one really bold scientist from Eli Lilly has changed the whole dynamic.
I'm a birder, so I have a unique perspective in that I can see the impact of the horseshoe crab, not only because of the horseshoe crab itself, but the species that rely on it, including birds and turtles and sport fish and all, anything that's in the ecosystem that it's impacting. And so we were aware at Lilly that there was an alternative to the LAL test reagent. We generated the data, but it took a lot of alignment internally, and then externally, we had to convince the FDA and other health authorities around the world. But we did that with our data, and we convinced them. We drew a line in the sand in 2016, and we said that all new drugs that are manufactured at Lilly, we would apply recombinant factor C to them. And so our first product was approved in 2018, and that was a milestone because that was a first in industry approval of any medicine using RFC for release. And then we've subsequently had 6 additional product approvals since then, including our 3 COVID-19 antibodies. Our goal when we set out was to convert all 8 of our manufacturing quality control labs at the time to RFC, and we've done that. And it's been, industry is following. We have our peers, Sanofi and Pfizer and Roche Genentech have all come out and made statements similar to what we've done, which is that we'll convert our water testing and all of our new product testing. So there's definitely a momentum towards more sustainable endotoxin testing.
One footnote on that. America truly is lagging behind on this adoption. Europe, Japan, Korea have all adopted the standards to approve RFC in testing. It's really our country that is the problem. The horseshoe crab has been around for 445 million years. It's survived 5 mass extinctions. And it's not until the last 100 years that humans have threatened it with extinction. I believe that when humans cause the problem, we have a responsibility to create solutions. If we look back 10,000 years, the world was a very different planet. There was much more biodiversity. But because of our human presence, we have actually closed out so many of those opportunities to let those species thrive. We're at a point in time where I think that by combining both conventional approaches to conservation along with new technologies and biotechnology, that we can actually envision a much richer, more bioabundant future. It takes time to make changes happen in conservation. The conservationists that we work with, in some cases, they have to work at the pace of nature. But the truth is, they all live in the long now, and we're all looking to see how we can build a better future. Thank you.
I've got about 3 hours of questions here. What kind and range of money would be needed to run a national biobank with samples of all the endangered species?
It's such a damn good question, Kevin, and it's one that I don't have an answer for. But I know it'll take an act of Congress to get federal dollars to do it, and we should get that through congressional dollars. I am talking to people at the national offices about this, of what it would take. So I'll tell you, the federal storage facilities are there. The databasing infrastructure is more or less there. The challenge of getting field biologists to actually add this extra step in their process, it's not a big deal. They actually like doing it. They understand the value. And in many cases, it's a one-off. I mean, they may get one at a time. The cell culturing with mammals, not a big deal. So, probably a million going into new research on cell culturing is definitely a number that is going to have to happen. And that could be for birds as well as other species. But I think the biggest challenge is actually getting the mandate to do it. And my goal is to actually get it written into these recovery plans, which the biologists have to do as part of their work. It's, this is their job in the U.S. is to protect these endangered species. And this is our American legacy if we do it right.
You give the example of where you've worked with other organizations to help accelerate acceptance and to overcome things that just seemed, what are, in the political realm, what are things that need to be done? Maybe not by revive and restore, but you said conservation takes time.
And that's correct. What would speed it up? I'm not so sure that it needs to speed up in some ways. This is working at the pace of nature. There's plenty of bureaucracy that I'd like to speed up, and but I think that it's actually been a kind of a healthy thing. When Stuart and I started this, we wanted to socialize these ideas, to have public dialogue about it, and I think that's been good for the culture. People need to actually realize how important these technologies can be and to integrate them.
A very long, nowish answer. Two closely related questions here. And I'm going to start with Jenny, and then I'll ask the second one, which is anonymous. Which conservation work is Revive and Restore currently partnering with, or which would you seek to partner with in the future? And part two of that is, how do you find and choose your projects and partners? And how do you support them? What are the ways you support them?
So probably the biggest bang for our buck of all of those projects is just enabling genomic sequencing. Some of those projects we may fund $50,000 grants, some of them $250,000, a few up to $1 million over 3 years kind of thing. But it's really getting these foundational tools into their hands. And that's something that is just going to get cheaper to do, and ultimately you won't need Revive and Restore to do that, because they'll be able, as a conservation organization or land stewards, to be able to just make that happen on their own. But for now, we're kind of a gateway for a lot of these organizations at that front. The bigger projects, like the engineering, really thinking about how to advance reproduction for birds, those are much bigger projects. Now, back to the question of conservation organizations. When we think about how we're making a difference, our goal isn't to try these ideas, move the science forward. It's actually to integrate it into a functioning ecosystem. And we are never going to be in a position to do that. We need the nature conservancies of the world to take those genetically resilient sea stars, captive breed them, and reintroduce them to the wild. That's a huge process that has to happen by other organizations, not us. So we're the kind of bleeding edge, try it, de-risk ideas organization, enable people to start to use these tools, and hope that they get translated out to, the big NGOs.
So when you're starting a relationship, you've been around for 10 years now, what does the arc of the beginning of the relationship, how do they typically happen, and how does it evolve, and when does it take off?
Well, we've done a really fun thing that we didn't know if it would work very well, but we called it, we started a program 3 years ago called Wild Genomes. And we asked for people to apply for research money to do sequencing on endangered or threatened species. But we put a very important requirement in it, that it had to be actionable, that it couldn't be just purely academic. It had to be something, information that once they gleaned from the genome, that they could apply in a way that it could actually help protect the species. And so, we've had way more proposals than we could ever fund. So we're looking for funding partners now as we build out that program. And so right now we have we just finished an open call around kelp ecosystems. And we did that in partnership with the Morris Animal Foundation and the Webb Family Foundation. And so they're kind of co-partners in it. And we actually have joint calls, and we have advisory boards that select the projects in a really good, healthy, functioning partnership. So I hope we can do more of that with funders.
Talk about unintended consequences.
Well, I have to talk about intended consequences because in the last 10 years, I always get the question of, we don't know about the unknown unknowns. And yeah, that's right, we don't. But we got to flip that question and say, what about the benefits of trying things? I mean, when I first started talking to coral biologists, and this was really early on, and we could see what was going on with global warming, almost everyone I talked to said, it is so desperate, Ryan. I mean, they were like going through PTSD diving. And they said, it's so radical, we don't dare do anything to risk the unintended consequences of meddling with the reefs. And that has really flipped in the last 5 years. Mary Hagedorn and all the scientists that we're funding, they're actually saying we have to intervene. We may not have to genetically engineer. We may take those corals that are doing well on one part of a reef, and we may translate them to another part where they could still do better, or we could, help with breeding and repopulating. But some of them are actually open to where the technology will take them in order to save the reefs. So I think it's the risk-benefit equation that has to be played out, and it just depends on people's tolerance, and I've seen that tolerance shift.
What is a tool or technology that seems almost possible that would make your mission easier?
Stem cell technology. I have a really good answer for that one. So one of the things that I'm learning, and again, this is kind of drafting behind human medicine, is that stem cell technology is changing everything in human medicine. I mean, what did I just read the other day? That stem cells were being used. These are cells that are taken from your own body that are then implanted to reduce inflammation or to help with Parkinson's, all these different applications. But understanding how to develop those stem cells has been incredibly hard. Now, they've never been used for conservation, not really. And so because of that, Revive and Restore is going to host this fall a workshop. And I've invited now probably 20 different scientists that are doing stem cell research in all different fields, from veterinary medicine. Stem cells are big with horses. I'm an equestrian, and I can tell you many high-performance horses get stem cell injections. They do these things first with animals, and they do it with prized cattle and prized horses. And then they work it down to humans. I want to work it the other way and work it for wildlife. So we're going to gather these scientists together, and we're going to do a format that I've used in other workshops, which is really interesting. We've done it now, I think, 4 different times on different topics. And the secret to the success of this, and it really has been a success, is we get 30, 40 different people from very different disciplines to all work over 3 days, night and day, in a retreat facility. And we give them 3 different challenges that they all buy into. They elect and opt in to different work groups. And at the end of the 3 days, they're supposed to identify the problem, identify the solution, develop a roadmap on how to get there, and develop what they think could be the translational partners to make it happen, and the funders. And then I have a mock group of funders who were present on the 3rd day of the workshop. And I can tell you, the night before, these teams stay up half the night trying to get their decks together. And they pitch it as if for real. And what has happened on numerous of these workshops, the funders stepped up. And incredible research has moved forward. One in particular on gene drives for rodents on islands. It was incubated at one of our workshops. And so I want to do that with stem cells because no one's paying attention to it. And it's the secret ingredient that will allow all of these different projects to move forward for engineering.
There was a question that Kevin, I peeked at it, but he didn't give it to me. But I remember it was someone asking, could this technology be used to stop invasives? Like,
Oh, yeah.
could we do something with the Florida python?
Well, God, I really wanted to do something. It's a huge challenge reproductively with the python. So the technology that exists that's been worked on with rodents is called the gene drive. And what a gene drive does is it perpetuates very quickly a particular gene selection through reproduction. And when that happens, you could basically create sterility. Now, everybody is very concerned about a gene drive for one species, for example, getting rid of rodents on an island by introducing only males, right? You could just get that selection through, and all of a sudden you've decreased your population. But what if that rodent escapes off the island and then affects the good rodents other places? That's the big fear. So scientists are working on trying to figure out ways to create both self-limiting gene drives, and they have lots of different approaches to this, so that that unintended consequence doesn't happen, even should an engineered animal escape. But to me, it's one of the most humane things we can do, because what we do for these species, like killing thousands of Burmese pythons every year in Florida, if we could create sterility and remove them, it would be a much more humane thing to do. Using rodenticides on islands has had huge mortality on other species, especially raptors who eat those sick and dying and dead rodents, and they ingest the rodenticide. So, working with invasive species is huge. Now, where they're making progress with this, because I need to give you more success stories.
Yeah, we should, just a little.
This is a good one.
Oh, sorry.
It's a good success story.
Okay.
So Hawaii had mosquitoes introduced, I think it was in the 50s. I didn't study that stat, but in the 1950s, 1850s. Oh yeah, they came in. They definitely came in with the whalers, and they came in and shipped ballast and everything else. Those mosquitoes should never be in Hawaii. There are 5 species of in Hawaii. Some that carry human pathogens like chikungunya and yellow fever and all these other things. But there's another horrific one that carries avian malaria. That's what those honey creepers died of. And it is causing extinction like nothing else. The big challenge is, as these island temperatures rise, the birds who used to seek a refuge at higher altitudes where the mosquitoes don't go, now they can't go any higher, and the mosquitoes are going as high as they can. So there's no mountaintop to escape to. So people in Hawaii have known this problem, and they love their birds, and they've been completely conflicted about what to do with it. This is the animal-human conflict that just keeps playing out. And so gene drives were proposed. I was one of the ones who did workshops in Hawaii, and they just went, GMO, forget it. But there is a solution, and there are folks that came out of Google X Labs that have used what they are calling an organic substance called Wolbachia that they can engineer into the mosquitoes that causes infertility in the mosquito. And also, because of their incredible AI expertise, they've been able to grow these mosquitoes in great quantity because they can use AI to sex select, something that, we haven't talked about the role of AI in all this, but it's just going to be really transformative for things like captive breeding of small insects and everything else. And Wolbachia is now being set for trials in certain islands. It's really worked its way through from, I think, having the scariest idea of a gene drive to something that now is being called organic, being passable. But at the end of the day, it is engineered, and it will not absolutely knock out the population of mosquitoes. It will just drive them down, and they're going to have to keep reintroducing these mosquitoes over time.
Some years ago, I was absolutely startled when E.O. Wilson confessed that if all the mosquitoes disappeared from the world, he would not object. I mean, is this biological extremism?
Well, look, I was a big proponent. I did a workshop, and I called it mosquito-free Hawaii. I wanted to get them all.
Well, getting off Hawaii makes sense if they were invasive,
Yeah.
But off the entire planet?
No, but it's complicated. Yeah. There, many species of mosquito. They're not all bad.
It is not a charismatic species. So Adam asks a question: with so many extinctions and populations in decline, how would you prioritize which species you focus on?
Yeah, well, so as an entrepreneur, I'm purely opportunistic. I look for passionate people. I look for science that's going to be really applicable, that can make a difference. Those are the biggest things that we look for. At the end of the day, when we put all the different wild genomes proposals together, we try to look at, is there any synergy between teams that we could actually, if somebody's working on one part of the coral challenge and another, could we put them together? We look for things that are outliers. The project One People, One Reef, and I have two people from One People, One Reef right here. Nicole Crane and Giacomo, who told you about his genomic sequencing. We look for people that are really making a difference and that are bringing us sort of a broader view of this whole challenge of integrating technology. And I think Magul did such a nice job in that very short video that he did from a very remote island at, I think, some ungodly early hour to help me with my talk. Weighing that whole challenge that they have between traditional knowledge and new innovative science, and how do they understand more about what's going on with their fish that they depend on. Because, typically environmentalists come in and they say, oh, you just need to create a marine protected area. You know, just wall it off. And you can't do that when people are living on it. But can you say, well, let's have science help us tell you how healthy that population is and at what density it needs to be, or that coral reef, what challenge they're having with their livelihood. But I think that that project in particular is profound in that it truly is collaborative. They are really learning from each other. And I think, you know, that is really my goal, not on traditional knowledge, but I gotta say, conservationists are very traditional. I know you're using it in a different way, but it is an old school approach. This, you know, wall off and protect is one school of thought. And they are completely anti-intervention. I mean, there were people when the California condor was going to go extinct that said, let them die with dignity.
Right.
I mean, David Brower, this dearly loved person, said, let them die with dignity.
They freaked out when they started using condor puppets to feed the condors. They said that would ruin condors forever, that they'd just become fast food condors.
And there are now 350 condors flying in the wild.
On that note, let's all give Ryan a hand. Thank you so much.
Thank you. Thank you, Paul.